Hello, everyone, and welcome to another webinar hosted by the CMT Research Foundation. I'm Susan Rudiger. I'm the co-founder and chief mission officer. I started the Research Foundation because I come from a family with 6 generations of CMT. I was first diagnosed when I was 2 years old. I'm 54 now. I wear leg braces regularly, and I'm noticing the progression of the disease in my hands and my legs and my arms, and believe it or not, beginning to feel it in my shoulders. CMT was first described in 1886 by Drs. Charcot-Marie-Tooth. And in 1991, the first gene was found that causes CMT. Okay, so it's been 139 years since it was first described, and 34 years since the first gene was found, and we still don't have any treatments. To me, this is just totally unacceptable. This is exactly why we started the Research Foundation in 2018. We want to expedite the development of treatments. In the 7 years since we've been in business, we've funded 32 projects, and 2 have led to clinical trials, with many more showing promising data. That's right, 2 clinical trials. Drugs developed by Novartis and Augustine are in clinical trials because of the investment and the expertise provided by the CMT Research Foundation. But let's be honest, we know that the success rate for the development of drugs is not exactly high. And we know that we need a large amount of promising therapeutics that can lead to clinical trials in order to realize approved therapies. And if we're fortunate enough to have early successes, if Novartis and Augustine make it to FDA approvals, we still can't stop. Patients need options for treatments. There are hundreds of mutations that can cause CMT, and not every person living with CMT today would benefit from the Augustine or Novartis drugs. So the CMT Research Foundation continues to invest in projects that have the potential to be successful. We invest in drugs that might have early promising evidence, but they were stalled or stopped due to technical difficulties. Our funding can help overcome these difficulties and help advance them into the clinic. We also invest in companies with technologies that could be relevant to CMT, but they haven't been tried yet. And this is exactly why we're here tonight. I'd like to introduce you to a company called 82VS, whom I met in 2023. When I met them, they had never heard of CMT before. They didn't know about the CMT project that was stopped by Ionis for safety issues. They hadn't heard about the success DTX had in overcoming that toxicity, and then how DTX had been acquired by Novartis for $1 billion. They listened to my story and they realized that their antibody technology just might help overcome the toxicity issues in a different way than Novartis's drug. I introduced them to our scientific team, and after several conversations, we all believed that this was a technology worth pursuing. So we funded their work. That's what we do at the CMT Research Foundation. We fund the science that addresses challenges in drug development for all types of CMT. We started the CMT Research Foundation to close the doors of the Research Foundation. And we will do this once everyone who lives with CMT has a treatment that slows, stops, or reverses the disease. We're fortunate enough to have a robust community of supporters, primarily those who have CMT or love someone with CMT. And we've been lucky enough to raise the money to have the ability to invest in these 32 projects. So thank you. Thank you to everyone who has supported the Research Foundation, either by giving a gift, asking a friend to give, or hosting a fundraising event. On this call tonight is Laura McNeil, the CEO of the CMT Research Foundation. She is also a fellow patient. She can count at least 3 generations in her family who suffer from the symptoms of CMT, and she herself is beginning to experience the effects of the disease in her own body now. We are founded by patients, funded by patients, and led by patients. Also joining us tonight is Dr. Megan Drummond. She's our Vice President of Research and Drug Development. She joined us from Regeneron, which is a mid-sized pharmaceutical company. There, she led the development of a gene therapy for people with another nerve disease, hearing loss. Dr. Drummond developed and dosed deaf patients and witnessed the success of them hearing again. Deaf people were hearing again because of a drug that Dr. Drummond and her team developed. Now she brings that expertise and drive to CMT. She leads our scientific strategy, She works with our volunteer team of scientists and drug developers to choose which projects to fund, and she's constantly looking for more partners to bring into the CMT world. Also on the call today, we have Dr. Carlos Loya. He's a venture partner at 82VS. He's a neuroscientist and a biotech executive with a proven record of success in starting and building innovative biotech companies. He has led their CMT drug development program that was in partnership with a research foundation. In a moment, Dr. Drummond and Dr. Loya are going to try a new format for our webinars. Instead of getting a lecture from Dr. Loya about his project, he and Dr. Drummond will have a conversation about the CMT program at 82VS. As they talk, if you have questions, just pop them into the Q&A feature. After they're finished, we'll take about 20 minutes to answer your questions that are there. Out of respect for your time, we'll close the formal session before 8 p.m. But for those of you who want to hear more, we'll stay on afterward to have a conversation with you. We'll do this by moving into a breakout room where everyone can go on camera and open their mics. If you're interested in joining that session, just stay in the Zoom meeting. You'll get an invitation to join the breakout room. Accept it if you want to stay and leave if you need to go. So let's get to our featured session. We're just about to hear how fast drug development can be when experts like Dr. Loya and his team partner with Dr. Drummond and the other scientists from the Research Foundation. Remember, we met the team from 82VS just 2 years ago. All of this work is possible because of our supporters. So please join me in welcoming Dr. Carlos Loya and Dr. Megan Drummond.
Thank you, Susan. That was a wonderful introduction, and I'm really glad that Carlos and I can present tonight. Also exciting to try our new format of where instead of, like Susan said, just lecturing, we get to have a bit of a conversation and talk to everybody about the things that get us so excited about science and solving CMT. So Carlos, I would like to start with, what about your conversation with Susan? Because I know just how compelling she can be. What about your conversation with Susan made you want to follow up and study CMT for your first program, or for the program we're going to learn about tonight?
Well, I think primarily because of how CMTRF is, as we've just heard, patient-driven and as well as mission-driven to develop medicines. And I think that is perfectly in line with the mission of both 82VS and, our venture studio, which is part of Alloy Therapeutics. So 82VS, we're an early stage biotech venture studio, and we're focused on asset-centric and capital-efficient biotech company creation. What that means is that we build companies that make medicines, and we let Alloy Therapeutics build the platforms. And just to give you a sense of what Alloy Therapeutics is, because they're really what power our novel genetic medicines and delivery platforms, it's a company that's both a platform company that has 6 different modalities around antibodies, genetic medicine, cell therapies, now delivery. And ultimate goal is to provide access of all of these different modalities to partners, whether it's a pharmaceutical, biotech company, a venture capital, a venture philanthropy. And we have now over 100 active drug programs. And over the last 6 years, we've demonstrated, our track record of success, where now 20, we're happy to say 20 of programs that are currently in the clinic were discovered by Alloy Therapeutics and our platforms. And we have the capabilities. We have 5 research labs around the world, 2 in the US, 2 in Europe, 1 in Japan, that allow us to really build drugs end to end. And the 82VS Venture Studio leverages all of these ecosystem, as we call it, as well as our pre-seed fund to bring programs, early stage programs forward. And, just to give you a sense of more of the modalities that we're actually leveraging for this partnership with the CMTRF. So we're leveraging our antibody platforms, which allow us to, with exquisite specificity and potency, target extracellular targets, which I'll hear in a moment, is very relevant as we look to deliver to specific tissues. Then we leverage our genetic medicines platform, which is a next generation antisense technology which now allows us to target genes inside of the cell or intracellularly, and then modulate them either to bring them down or downregulate them, or upregulate them through different ASO mechanisms. And then we're bringing those two pieces together in our delivery platform, which then allows us to take oligonucleotide genetic medicines to tissues where it's difficult to take them without delivery technology.
So one of the things I really love about the approach of 82VS, Carlos, is just how versatile it is. I mean, you've got genetic medicines here, you've got antisense oligonucleotides, which we've heard about for other indications and other things. And, we know about antibody therapies and how powerful those can be, and also that they have a pretty strong safety profile because of the so much specificity that they can give you inside of the body, right? In terms of being very specific in what they target and how they target. I think we often talk about the approach of, or the question, the big question for CMT around delivery, but delivery can mean so many things, right? And so,
Mm-hmm.
could you tell us a little bit more about exactly what that means to you, and what is a delivery hurdle, and how are you guys starting to address that?
Yeah, so I think you put it just right. The challenge of delivering this medicines, the solutions to um, many patients, particularly with CMT, is the fact that the human brain, the human body has many physiological barriers that are evolutionary there for really good reasons. We have the blood-brain barrier, which keeps toxins and nucleic acids, viral particles from the brain. We don't want them there. That's a good thing. But it prevents medicines from getting, if they're in the form of an antibody or an oligonucleotide, into the brain effectively. Similarly, in the periphery, we have similar physiological barriers that we call the blood-nerve barrier, which are allow for the peripheral nerves to have balance and homeostasis between the neural compartment and the periphery. So we also have to develop new technologies so that we can deliver genetic medicines to this peripheral nerve tissues, and that ultimately will add, the specificity, the efficacy, and also importantly the safety to the overall drug, and also ease of administration, because there are ways to deliver genetic medicines and oligonucleotides into the peripheral nervous system or the brain using intrathecal delivery. So what that means is a spinal tap into the spinal column, which is very invasive, has its own kind of safety issues. So if one can develop a more systemic form of delivery, either subcutaneous, or similar to how type 1 diabetics, they inject insulin subcutaneously or intravenously, that allows for a much more systemic and also convenient way to develop a medicine. So that's what we're doing as part of this partnership. We're doing what we like to say a new kind of CMT therapy, where we're taking, similar to, I like to use the analogy of, Amazon trying to deliver a package to your home. Of course, it uses delivery vehicles, and it needs an address to actually deliver it to your home. And that's what our antibodies are providing. They're the delivery vehicle with the right kind of address to, take it to its right location. And then we bring the medicine package in the form of a genetic medicines that then can have what we expect is a disease modifying effect. And within CMT, I think, as Susan alluded to, we understand the genetics of many CMT diseases. There's over 100 genes that are associated with CMT. We're first starting with you know, CMT1A and PMP22, but the important thing is that with oligonucleotide and genetic medicines, we can actually do a lot by modulating both, the but the levels of PMP22 by downregulating it, or other genes by also downregulating or upregulating them as potentially needed.
So what kinds of, you mentioned antisense oligonucleotides. Have those been used in the clinic yet? This technology that you're talking about for how we can regulate PMP22, are there some examples of that?
Yes, and that's why we believe oligonucleotides are the right modality for CMT. One, because oligonucleotides, they're already a FDA approved modality with multiple, now 17 FDA approvals. And what's most interesting to our conversations is that in the last 5 years, there's been 7 approvals, and that's been largely driven by the discovery of a delivery technology. This for hepatic cells. It's called the GalNAc technology, which is conjugated into the oligonucleotide and enhances its delivery into hepatic cells or to the liver. And as you can see, that has led to multiple approvals in a very short period of time. And now I think that there's, we understand the power of this technology. And I think now what we need to better understand is how to enhance its delivery to other non-hepatic tissues or to organs that are not the liver, such as the peripheral nerve.
Hey, and I was familiar with GalNAc and the GalNAc technology and how impactful it was. But until you put the slide together, and I saw it laid out this way, right? That the first one was in 1998, and then it took us until 2013 to get another ASO target to be delivered, right? And then was sort of creeping along, and then boom, there's GalNAc, and now there's lots of liver targets. It seemed like that really helped to open up the field and allow things to move quickly. So what do you think is what's the peripheral nervous system's GalNAc?
So, we alluded to that earlier. I think the modality that we're going to use is antibodies. And I think right now what we're starting with is the transferrin receptor as the initial target. And we believe this is attractive because, one, it's well validated as a delivery shuttle. Transferrin receptors and the transferrin kind of system is our primary carrier iron to many tissues. So it's expressed at high levels in many tissues of interest, one of them being the peripheral nerve, the brain, the skeletal muscle, so many relevant tissues. And also, it's important that it carries iron quite rapidly, sometimes in some cells, 500 molecules per second. And that's important because we want as much of the medicine to get into the cells as possible. And as I mentioned, it's validated. So it's been shown by other companies to effectively deliver oligonucleotides, either antisense oligonucleotides or ASOs or siRNA into the brain, deep into tissues in the brain where otherwise it wouldn't effectively be delivered, the peripheral nerve, as well as skeletal muscle. And now we're seeing with some companies like great signal, particularly in skeletal muscle by leveraging transferrin. Many very debilitating musculoskeletal diseases now are being shown to have clinical effective treatment. So not all transferrin antibodies and shuttles are created equal. Some actually, depending on their properties, get better biodistribution to the muscle or the brain or other tissues. So that's what we are doing. That's why we actually started by you know, from essentially from scratch to develop a diverse set of antibodies that allow us to, test multiple profiles to see what gets delivered best into the peripheral nerve.
That's really cool. And it sounds like I love the analogy of the Amazon truck and having the right address. And it sounds like we've got potentially the address to the peripheral nervous system, right?
Yes, yes, that's the ultimate kind of hope and the hypothesis. And right now we're testing it.
So we've talked a little bit, you mentioned the antisense oligonucleotides. And so now we've got this transferrin receptor, the antibody, conjugated to an antisense oligonucleotide. So that's what you're calling the AOC, right? The
Mhm.
antisense oligonucleotide conjugates. So
Yeah.
can you tell us a little bit, like, can you dig a little bit deeper into that and maybe talk to us about why CMT is such um, a good first exploration for this technology that you're developing?
Yes, and I think Susan, I think laid it out beautifully where we already know, and since it's been 30 plus years since the first kind of genetic association, um, first gene identified that was associated to CMT. Now we have over 100, and we understand overall the mechanisms of action. Many of them are gain of function, which means we need to downregulate that gene. Some are loss of function, which means that we may need to upregulate the gene that is still being expressed or do gene replacement. Um, some are splice defects, and all are located within this peripheral nerve, whether it is in the kind of peripheral Schwann cell compartment or neuronal. So that's what we're trying to do with the development of an antibody oligoconjugate. We're looking to now develop a vehicle that will allow us to then plug in different different types of ASOs that are targeting different genes for potentially treatment of multiple CMT types. And of course, we're, as I mentioned earlier, starting with CMT1A.
That's great. So we've thrown around the term ASO or ASO. What can you tell us a little bit more about like the biology and how that actually works to alter DNA expression or expression from DNA?
Yes. So I'll just quickly run through kind of also what we're doing at Alloy to enhance the properties of ASOs. So the foundational antisense technology was started, decades ago, about 30, 40 years ago. And many of the drugs that now are FDA approved are using this two formats, which we call the gapmer, which have both DNA and RNA in one single-stranded stranded nucleic acid, or they're steric blockers, and they only contain modified RNA. The gapmer is able to downregulate a gene. Steric blocker can do multiple things. It can do splice modulation, and that's behind one of the seminal drugs that has been FDA approved, nusinersen, which is a drug for the treatment of spinal muscular atrophy. And what we've done at Alloy is we actually are working with the inventor of the technologies that I just mentioned, Sudhir Agarwal, and because we know what are some of the challenges of those technologies. We know it's many times potency, specificity, and also in some ways the mitigation of inflammatory responses. So we've developed new formats. One of them being what we call here the anticlastic cyclic format, which essentially is, as it's seen here in this illustration, it's a cyclic antisense, not necessarily a fully linear antisense. This creates this transient cyclic structures, which give it added potency as well as specificity, and it mitigates a lot of the inflammatory responses because of its structure, because we are masking a lot of the elements that interact with receptors in cells that activate the inflammatory responses. And normally that's not a good thing, that or one thing that we're looking for when we're making a drug. And we've done many tests with our technology. We've benchmarked against, over 20 clinical or FDA approved antisense, where we just take exactly what's right now being tested in humans, we reformat it into our technology, and then we see, and this graphs show that kind of head-to-head of the let's say clinical or FDA approved ASO when it's then reformatted into our technology. And when the curve goes down and to the left, that means it's more active and more potent and can engage the gene knockdown more effectively. And we see normally, when we've seen throughout kind of the span of new technological investments in ASOs, 2-3x improvements in potency. Here we're seeing kind of meaningful orders of magnitude, which gives us a sense of, okay, if we're more potent, then we could potentially have what is called a better therapeutic window. So the window between efficacy and a safety signal. So the wider we can make that, I think the more comfortable we're going to be. And we also have tested this, in inflammatory assays. And this is, for example, an actual approved antisense. And this assay is to show its inflammatory activity. So if the curve goes up and to the right, that's more inflammatory activity because you're releasing more of this inflammatory agent. Here is IL-6. And when we take that same exact drug and reformat into our technology, we don't see that response. So we're mitigating that. And ultimately, we're hoping that we're able to just have a more potent and safer drug overall. That's the overall goal of that technology.
I mean, from these graphs that I'm looking at right here, the inflammatory response you've really made a huge impact there, right?
Mm.
Compared to traditional technologies. This looks really promising and really fantastic.
Yeah, and now the goal is to do something that is also focused on now a CMT specific gene.
Great. So you're combining a delivery mechanism, which is using this antibody, right? Which is like the delivery truck now that has the right address for the peripheral nervous system. You've got the cargo inside of that Amazon truck, which is your anticlastic antisense oligonucleotide, which is a short stretch of DNA in this special configuration that reduces the immunogenicity, but will still function to reduce the expression of the gene, like in
Mhm.
this case, PMP22. And so it seems like this is a very promising approach to move ahead. Can you tell us a little bit about where your work is today and some of the things that you're looking forward to next?
Yes, and so as I mentioned earlier, so Alloy, we leverage our platform technology and antibodies, as well as our significant know-how to develop a fully human panel of transferrin antibodies, and we've characterized them and looked at many elements that are important for transferrin shuttle uh, function and safety. One, we don't want to, for example, inhibit transferrin activity. So that's one thing you have to always kind of screen out. And one thing that you need to screen in is, well, an antibody that's able to internalize into the cell effectively and engage the transferrin receptor with very high affinity. So we've now identified and and and, and prioritized 4 lead transferrin antibodies that now we've tested its biodistribution and seen that it actually does have significant uh, uh, efficacy in the brain and in the skeletal muscle. And we're now developed antisense oligoconjugates. We've done the chemical conjugation. We've tested in vitro and shown this activity. And now we're testing in vivo in humanized transparent models. And we're now, I think in 2026, we'll have validation of this technology and its ability to engage genes in the peripheral nerve. So for one quick kind of data point here, where we looked at the peripheral, at the distribution of this transferrin antibodies, we conjugated them to a fluorescent molecule that, as you can see here in pictures, where we can see the fluorophore, and we also actually compare them to another clinically validated transferrin antibody. So we're doing kind of that comparison. And as you can see, when we look at the brain and the skeletal muscle, we see that our antibodies are able to effectively, distribute into the brain and to skeletal muscles. And so that gives us the first signal that, okay, we're in the right direction. We have transparent molecules that work, and now we're doing the experiments to test that we can deliver oligonucleotides into the peripheral nerve, and that's what's ongoing. We are, as I mentioned, in the next in 2026, we'll have, the in vivo data readout in fully humanized transferrin mice. And then the goal is take from this 4 antibodies that we're testing, and we're testing multiple chemistries for conjugation to the oligonucleotide. And we're also testing, how much actual cargo we can fit into the delivery truck. And so we're testing different, levels of antisense that we can link into those antibodies. Then we're going to pick the preferred format for the peripheral nerve. And then we're going to conjugate PMP22 antisense that we then are able to now validate in the same in vitro and in vivo systems, with the ultimate goal to now do a full screen and find the most drug-like, most potent and safest PMP22 antisense that then we can also test in kind of the validated translational models of CMT1A. And I think that is kind of what we're looking for 2026 and early 2027.
I had muted myself based
Yeah.
on the data that you showed. Carlos, it looks really exciting in terms of what we can look forward to. I mean, you had those small pieces of tissue that showed that where it may normally go to the muscle, now we see like the little bits of neuronal tissue lighting up, which means that you're getting a lot more there and a lot less where you would normally expect to see it, right?
Yes, that's right. That's right.
Yeah, I really can't wait to see the data coming from the mice. I mean, to see that it works in cells, and then to see, if it's going to work in the mouse model, is really pretty exciting. So PMP22 is the first target you've talked about, right? Which is for CMT1A. And can you tell us, for patients or for broader patient community, one of the questions that we often get and something we really like to think about is, this is great for patients with CMT type 1A, but acknowledging that there's lots of subtypes of CMT and that, trying to find wins that will work across the board. Do you think this platform could be applied to other subtypes of CMT besides 1A, assuming success, which I'd love to do.
Yes, of course. I think that there is, and I think we've alluded to that. I think many times, and it'll has to, I think, be predicated on what can these genetic medicines do. And ASOs have now been validated, and we've validated our technology that it can do multiple things. One is degradation of target RNA. So that's what we're doing with um, the PMP22 and CMT1A. And there's other ways that you can correct a pathological genetic variant. There's splicing variants that are associated, or splice uh, uh, aberrations that are associated with some CMT types. We can use splice modulation, and in those particular chemistries, there's also ways to upregulate genes if it's a haploinsufficiency. And, there's other ways that one can even edit RNA using ADAR. And all of these could have and play a role in future medicines. And I actually see that the CMTRF is funding many of those, projects where you're looking at ASOs for splice modulation or for other mechanisms that are not just knockdown. So I think that that's the goal. And, we are, as I noted earlier, a organization that prides itself in providing access to that technology. So I think that is one piece where in it's often, and we see it, that these shuttle technologies get siloed within certain organizations and certain companies, and that prevents the use of those delivery technologies or other genetic technologies from being more broadly used. And well, our goal is, once we have, and I think, done our necessary validation, I think the goal is to provide access to this technology accordingly, so that others can also make medicines for other CMT types.
Great. Thank you so much. I think we're running short on time. It's been really exciting to hear about all of this. So I think we're going to switch it over to Susan to take some questions from our audience.
Thanks, Meg and Carlos. This is so fun. Carlos, you are definitely a scientific wizard. There's no doubt about it. And I appreciate the very deep conversation that you're having. We're getting some questions about some of the things that you said from a technological, like a technical perspective. So one of the questions we got was, you said that an ASO had been used before in a disease with muscle atrophy. Can you explain that a little bit better and what you mean and why that's important to your drug? You were
Hmm.
talking about nusinersen, I think.
So there have been multiple, I would say now, validations clinically, and now that are FDA approved for antisense. I think with respect to nusinersen, so here is a disease, as it's called, spinal muscular atrophy. And this particular uh, medicine is delivered intrathecally, and it's targeting a gene called SMN2. And what it does, it actually does splice modulation, where it creates a functional form of the SMN protein that then corrects the deficit of a analogous protein called SMN1. And that is I think very relevant, because it demonstrates that this technology is 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 very potent. It can um, you know provide, and I've been, uh, I've seen I've been very privileged to see children that have, you know, would have a prognosis of a few years of life to be climbing a wall at age 5 or 6 because of these drugs. And so that's what I think is very relevant. Now, again, going back to the previous point of well, intrathecal delivery every 3 months, not ideal. But of course, you know, I think everyone, every child and their parents and adults with SMA will certainly, have agreed that, hey, it's worth it. But I think that there's there's more we can do, I think, to better deliver this genetic medicines.
And so spinal muscular atrophy is a completely different disease from CMT. It's more of the central nervous system than it is of
Mm-hmm.
the nervous system. So
That's
how
right.
does the delivery vehicle need to change from central to peripheral? Like, I mean, how do we change the address from the spine to the peripheral nerve, so to speak, to use that analogy?
Yeah, that's a great question, and it's something that we're trying to better understand. And it's something that, the rules haven't been kind of perfectly written yet on, what is the perfect address for TFR if you want to take it to the brain versus the muscle versus the periphery. But we know what we need to tune. We need to tune its affinity to the transferrin receptor. We can tune the size of the molecule. Um, we can uh, tune its internalization, and all of those different pieces, I think that groups have seen, changes its biodistribution, and now, I think our goal is to figure out empirically what is the address for the peripheral nerve, but it's not something that we can, say a priori or before kind of we do the experiments.
Sure, sure. And so I could understand that a drug that maybe could get to the peripheral nervous system could also get to the liver or to other organs and
Hmm.
might cause some real toxic side effects. Um, if you do get the address right to the peripheral nervous system, do you anticipate? I know this is kind of a not very fair question, but I'm going to ask it anyway. Do we anticipate any off-target effects or what the general community would think of as side effects that we wouldn't want in a drug?
Yeah, it's a very difficult question before, we actually do the studies. I would, you know, with respect to PMP22, we know that it's expressed in other tissues. It's expressed in the liver, it's expressed in, in other areas outside of the peripheral nerve. And one thing that we can always do is test for those off-target effects in first, in small animals like in mice, and then we also test for off-target effects, that could be on target, as in engaging the PMP22 mechanism, or complete off target, where it's like we're not even engaging PMP22. So that's why I believe that genetic medicines and ASOs are well suited here, because we reduce those off target effects quite well. Um, and then the other, I think, evidence we see is from some of the DTX and Novartis. We know that it's in the clinic, so it's gone through rigorous clinical or preclinical safety testing, and that is also a systemic drug. It's IV dosed, so I think that there's a good, okay, I would say, uh, precedent for safety, and I think now the potential goal is, hey, can we improve upon kind of the both delivery and route of administration. So, for example, can we make it subcutaneous? That would be a really, I think, significant improvement. So I know that I'm not answering the question because I think
It's not a question
it's
review.
impossible
Yeah.
without the data. But I think that we can piece a good argument for why engaging this particular gene will have a good safety window.
Yeah, thank you. One of the things that's become really evident from the questions that are being asked is we've got a lot of scientists. I think we've got some patient scientists in the room, just looking at some of the names, and then we've got a lot of people who are interested in working in CMT who've come, which is exciting for me as a patient because a webinar like this opens up to more scientists who want to come and learn and possibly get involved. So thanks for being here and giving a scientific talk as well as a patient-friendly talk. It's exciting to see. One of our questions from one of these scientists is, tell me about the dominant loss of function versus a toxic gain of function. And Meg, if you'll take a second to explain that to the patient community who are not scientists like me, and then Carlos, if you'll answer the question about how we think they could be relevant to the different mutations in CMT. So Meg, will you give a little primer on the loss of function, gain of function?
Sure. Yeah, so when we talk about genetics and when we talk about certain mutations, there's a couple of terms that scientists like to throw around that have a lot of meaning. One is when something's dominant, that means you only need one copy of the mutation in order to see the disease. When something's recessive, that means you need to have inherited a mutant copy from both parents, and so you have two mutant copies of a gene. And so then when you think about, let's go back to the dominant scenario, when you just have one mutant copy. Now, that mutation can either lead to what's called a loss of function, where you're losing 50% of the function that your cell would normally have that can support the healthy functioning of that cell. Or sometimes rather than it being a loss of function, it means it can take on a new function that can be toxic in the cell. And so there's this nuance between, is it that you've lost some function, 50% of your function, and that's not enough to support health? Or is it that you've got 50% would be enough, but now you've got this toxic taking on a new property and sort of going haywire that's happening? And so, Carlos, it's a great question from the audience. And, as you think about ASOs, could you use an ASO to address either the dominant loss of function or a toxic gain of function, or is it one or the other?
I think it's both, but it requires different mechanisms. So let's start with the toxic gain of function, because I guess the toxic gain of function is analogous to what is happening in CMT1A. Well, you have duplications, and that overexpression does lead to the pathology. There's also the situation where there's, as you noted, we all carry two genes or two copies of every gene, and there could be one variant that is expressed the same as the normal variant, but this one is toxic. So one thing that we could do is just specifically target the variant with the mutation, and you can do that now. And we've shown this with particular anticlastic technology that we can, with very kind of exquisite specificity, target one nucleotide change in a string and specifically target the toxic allele and not touch or not significantly touch the normal one. Now, I think that's something, and it's being done already. I think we're seeing it being used in other indications such as Huntington's, for example. Now, dominant loss of function is more complex because now you're talking about, okay, you have lost one of your normal genes and you're only expressing one, and that's not enough. So you need to bring that up. And that's now actually been shown with ASOs to be effective in the clinic. You see also Novartis recently acquired a company called Regulus, and Regulus developed an ASO that can target specifically a microRNA that inhibits the expression of a gene that's part of a haploinsufficiency, so or the dominant loss of function here. And you see pretty dramatic clinical benefits. This also been seen in other rare pediatric epilepsies. So we're able to do that with ASOs, and I think now that's been clinically, I think, validated. So that's why I showed kind of that slide with all of different mechanisms, because I think there is potential solutions using ASOs or siRNA, for that matter, to treat different genes that impact CMT. So it's actually really important to know and do what CMTRF is doing, which sometimes it's like, okay, can we learn what the biology is behind this particular CMT type? That is critical to then inform what is the strategy to use to develop the genetic medicine.
Mm-hmm. I want to encourage the patients, maybe the non-scientists on the webinar, to ask some questions. Please know that there are no stupid questions. I'm not a scientist, and when I was first figuring out CMT, I had a lot of really stupid questions because it's not my language. I wasn't raised as a scientist. So we welcome all questions, and please don't feel like you're asking anything that's stupid. We want this science to be understandable to you. So please ask the questions that you have. While we have a moment to ask those questions, Carlos, I have a question for you. Why are you excited about CMT as an indication? You've named a lot of other drugs that, or I'm sorry, a lot of other diseases that might be easier for you guys to tackle than CMT. Why did you decide to get into CMT? We met, and then you came to our convention, you sat and listened, but what about CMT enticed you? What brought you in?
I think there were, well, multiple moments, but I think it all was catalyzed by our first conversation. And then being able to, one, see that, there is very deep understanding of the causes of CMT. And, the fact that we have potential technologies that can be leveraged to making those medicines. So I think that is, from a drug discoverer perspective, I think very important. Then the other one is, I think, quite honestly, the engagement of the CMT community. And I've had the privilege to attend both the CMT convention in Cambridge as well as other CMT patient forums where, it's really, I think, important to see that level of engagement, because ultimately that leads to change in not only drug discovery, but also in, I think, which is really important later on in regulatory changes within the FDA, so that one can potentially have a better understanding of what's the path for clinical development and the ultimate approval. Then I think thirdly is, I think DTX did a lot of great work, and its partnership with Novartis, I think now clearly validates that there are pharmaceutical companies that are looking to help uh, the community develop medicine. Because I think one thing that maybe was holding back other companies from moving into CMT is that there wasn't really potential clarity on the regulatory path, or potential interest from larger pharmaceutical companies. Because I think it's all it's important to have kind of a I think a rich ecosystem where you have early stage companies and pharma companies and investors all working together, and academia and patient organizations working together to make a medicine. And now I think that was an important, element that I think has catalyzed many other companies to raise the funding that they need to get into the clinic. So I think just kind of maybe putting it shortly is kind of one is you, the CMTRF, uh, and the patient community, and also the great work that's been done by previous companies like DTX.
Awesome. Well, I can tell you we're a motivated group of people. 135 years since knowing about the disease, and we still don't have any treatments, and we're watching generation after generation after generation of families and their bodies deteriorate. So it's, when you see in the news that SMA and Huntington's and sickle cell and this whole litany, Duchenne muscular dystrophy, are all getting these approved treatments, and we still are at a goose egg for CMT. We're super motivated. So very, very honored to have earned your attention, and have you guys focusing on CMT. I think the $64,000 question that everyone wants to know, which is also probably not fair, but I'm going to ask it anyway, is given what you know today, and let's just assume that everything works well for 82VS on time. We don't have any miracles, but everything looks like it's going to be successful. When do you think you might dose your first patient? That's such an unfair question. I'm going to ask it anyway.
Well, first, it's my honor to be here. So really appreciate Megan, Susan, and the entire CMTRF team to have this opportunity. Um, I think with respect to preclinical development, particularly antibody conjugate development, uh, if we look at the benchmarks for drug discovery, I I think we we see that the first the identifying the right shuttle
Mhm.
is going to be important, and I think that's our mission for 2026. Identifying the right cargo is also critical. That's the mission for 2026, 2027. Bringing them together, and then now putting them forward for uh, translational mouse studies, as well as the necessary manufacturing to move it into non-human primate. That will take another 1 to 2 years, and then the necessary talks and clinical package. Those things have much clearer timelines, which is 6 months to 9 months. Kind of that's usually what the FDA looks for, and or other regulatory bodies are looking for in order to start dosing. And then I've had the privilege to work on now 6, 7 clinical programs, and then uh, kind of that usually getting into the clinic, once you actually have the right safety package, is kind of it you know do it as quickly as possible, and with a community as engaged as CMT. I think recruitment, I I think will be benefited by that, and also we'll learn a lot from Novartis and
Hmm.
their trial, because I think just look at that timeline, you know, from the moment that, DTX discovered kind of their development candidates to now their first dosing. I think if you look at that time frame, it will give you a good sense of kind of when and where when we can get to the first dose patient.
Yeah, I mean, we met DTX in 2019 at Bio, and here we are in 2025, and they're dosing patients. So 6 years from idea to first patient dosing. It's exciting,
Yeah.
and they're paving the way for so many others. So I'm looking
Hmm.
at it's 8 o'clock now, and I promised people that we would wrap up the formal part of the webinar at 8 o'clock. There's still a number of questions in the chat. So if you popped a question in the chat and you want to come on and have a discussion with Carlos or Meg or myself or all of us, you're welcome to stay once we get that link up into the Zoom. But I just want to take a moment to thank you, Dr. Loya and Dr. Drummond. You guys it's so clear. You know exactly what you're talking about. You know what you're doing, and you seem to have a lot of hope and promise, a lot of optimism for CMT. We're really grateful for that.